Hello everyone, welcome to my channel. Today we are here with a new topic of pharmacology. Now we are going to study a very important topic about clinical trials. So now before studying this topic, we need to have some basic concept. If we see till today's date in market, there are thousands, millions of drug available in market. Now the question is how this drug came into the market. Like suppose in my laboratory, I am doing some research and discover one new drug. According to me, it's a very good anti-hypertensive drug. So what I did, I made 10,000 pills and took it to the chemist and said, please sell it. Can I do this? Obviously, the answer is no. Why I can't sell? Because it is a chemical compound. It can cause any harm. If somebody dies, then who is responsible? So obviously, before any drug come into the market and can be sold, there must be extensive testing on the drug. Basically, we have to test two things. First is the efficacy, and second is the safety. We need to know the drug that we have discovered is effective or not. Also, we need to know the drug that is safe or not. So, what we will do, we will check in animals. We will give this drug to the animals like a rat, rabbit, mice, guinea pig. So, we will test them to see how effective it is. Basically, efficacy is animal models. Model means that we first induce a disease to the animals. Suppose if we have to test an anti-hypertensive drug, so first what we will do, we will make a rat hypertensive. So there are lots of ways to make a hypertensive. If we perform any experiment or test in animals, it is called as a preclinical studies. But if we perform the test or experiment in a human beings, it is called as a clinical studies. As you know, in today's date, animal studies, how much they are regulated. So there are many regulations. So in preclinical studies, we are not supposed to take any animals and give drug to anyone by our wish. We need to follow our proper guidelines, and the guidelines are given via CPCSEA, Committee for Purpose of Control and Supervision of Experimental Animals. So this committee will give us a proper guideline which animals we will take, how many animals we will take, what can be done, what cannot be done. So all these guidelines are given. So under CPCSEA guideline, we will do a preclinical studies. So let's assume we did an experiment in animals. It may be a rat, it may be a mice or a guinea pig, and we got a very good result. And we saw that the blood pressure has been reduced, and we confirm that drug is effective. And along with this, we need to see the safety as well. For safety, we will check the blood profile test, we'll check a kidney, we'll check a liver, we'll check a heart, and we found that all the organ is safe. So that confirm the drug is effective as well drug is safe. So what we did again, we made a 10,000 pills we have taken to the chemist and said, hey, now you sell it. Can I sell now? Obviously the answer is no. Why we can't sell? Because we have tested this drug in animals, but we have to give this drug to the human beings. So it is not guaranteed that the result shown in animals is going to be the same for a human being as well. So that means we have to study first on a human beings also, and it is called a clinical trial. So clinical trial is a testing of a drug in a human beings. So clinical trials are also have some guideline called as a good clinical practice guideline, GCP guideline. So in clinical trial, whatever we are doing or wanted to do is need to do under the GCP guideline. So one important questions can be framed here that the good clinical practice guidelines are not applicable to which studies. It is a animal studies, because for animal studies we have a CPCSEA guideline. So for this what we will do, we have to study in a human beings. It is not like that you can take any human beings according to your wish. Before you go for clinical studies, you need to have some license. For this there is a licensing authority. By name itself you know that authority that has a power to give a license. So the licensing authority will decide to give a license or not. If you get license, you can move forward for a clinical trials. So depending upon the country, there are different licensing authority. For example, for United States, there is a FDA and it's a worldwide. But if we talk about India, the India's licensing authority is a DCGI, Drug Controller General of India. This work in a CDSCO, okay, Central Drug Standard Control Organizations. So for Nepal, we have a DDA, Department of Drug Administrations as a licensing authority. So depending upon the country where you want to do a clinical trial, you need to take a license. So we can say this licensing authority is the organizations that looks at the clinical trials. So for getting license, you need to put the application called as a INDA, investigational new drug application. So along with this application, you will submit all the preclinical studies report to the licensing authority. They will review this report. If you can convince to the licensing authority that your research might give a potential positive outcomes, they might give approval for the studies. If permission is granted, then we go for our studies in humans. And the clinical trial has a many phase, and the important phases are a phase 1, phase 2, phase 3, phase 4. So now we'll see the phase 1. So in phase 1 trial, what we will do, here we'll take a healthy human volunteers. So now question is, we are supposed to give a drug to the patient, but here we are recruiting a healthy human. So what is the logic behind testing a drugs in a healthy people? Do you think the healthy people won't have a side effect? Definitely, there might be a risk of a side effects. The basic logic behind taking a healthy people is that a healthy human have a good immune system. Suppose if any drug can cause side effect, the healthy human can tolerate easily. But if already people have a weak immune system, in case if drug can cause any reactions or side effect, the person might cannot tolerate it and might cause a serious problem. So that's the reason we do clinical trial phase 1 mostly in a healthy human being. But some drug has a very toxic effect, like an anti-cancer drug. We know that the anti-cancer drug mostly cause a bone marrow suppression. They might affect a healthy person as well. So this drug, which we already know they are very toxic, we avoid testing in a healthy people. So the next question is that what we mostly cannot do in phase 1 clinical trial. The answer is we cannot test the efficacy in a phase 1 trial. The simple reason is that in phase 1 trial, we are taking a healthy people. Suppose we have a new drug, for example, anti-tuberculosis drug, but we have taken a healthy people. If person does not have a TB, then how we will know that TB is cure or not? So it is very important to remember that phase 1 is not for efficacy. If it is not for efficacy, then for what? So obviously it is for a safety. So what we do in phase 1, basically we call a healthy man. We give a drug and we'll see whether drug is safe or not. So let me ask the next question. How many humans have received this drug before? Obviously the answer is 0, because the very first time we are going to give a drug to the humans. If we are giving the drug for the first time, how we will know how much dose should be given and who will tell? So this is what we determine in phase 1 trial. Basically, we have already conducted studies on animals, so we already know what dose was used in animals. So using that, we can apply a formulas to derive the human dose, which is called as a human equivalent dose, meaning that the dose that is equivalent for a human based on animal studies. However, this dose fell within a range, say 50 to 500 mg. Now the question is, should we give a 50 mg or 100 mg or 500 mg? Who will decide this? So this is determined in a phase 1 trial. Essentially, we invite our healthy volunteers and give them to the lowest dose in the spectrum. Let's take example 50 mg and then conduct a extensive testing. We'll test a hematology, we'll check a RBC, we'll check a WBC, we'll check a kidney function test, liver function test, ultrasound, CT scan, everything we'll do. So if no issues arise, we send that person at a home. So next again we invite another healthy volunteer and give a higher dose, example 100 mg, and repeat all the tests. If there are no problems, we move to the next volunteer, increasing the dose further. So this process continues until an intolerable side effect appears. So this process continues until an intolerable side effect appears, which means the dose cannot be increased beyond this point, and this is called as a maximum tolerable dose, MTD. So the main aim of phase 1 trial is to determine the maximum tolerable dose, the highest dose that can be safely given. So beyond this dose, no further trials will use a higher dose. After phase 1 trial, we determine the maximum tolerable dose. Let's take example 500 mg. This means that in all future trials, no dose will exceed a 500 mg. It will always be lower. Okay, so the phase 1 trial helps us to determine the maximum tolerable dose, ensuring the future trials remain within the safe limit. So now we'll move to the phase 2 trials. So these phase 2 trials are conducted in two parts, that is a 2A and 2B. 2A trials are known as a proof of a concept trials, whereas the phase 2B trials are known as a dose ranging studies. Let's see about the phase 2A or proof of a concept trials. So the main purpose of a proof of a concept trial is to test for the first time whether the drug is actually effective for disease or not. So these studies provide initial evidence that drug may be effective. So in phase 2A, we primarily check the two things. One is safety and next is efficacy. So here is one important point about the phase 2A trials, that the first time efficacy is checked in the phase 2A trials, as we could not assess the efficacy in phase 1. So along with efficacy, we also check safety as well. So in phase 2 trials, we enroll patients, not a healthy volunteers like in a phase 1. So the number of patients is very small, that usually we take about 20 to 200. So these trials are often unicentric, meaning they take place at a single hospital or a clinical site. We are not taking patients from a different location or different countries. So after conducting a phase 2 trials, we determine whether the drug is safe and efficacious in a small number of patients. So once we complete a 2A trials, we move to the phase 2B trials, which is also known as a dose ranging studies. So once we have evidence from a phase 2A, we proceed to the dose ranging studies in a phase 2B. Here we adjust the dose either by increasing or decreasing it to find the most effective and safest dose for the patient. So what happens after phase 2 trial? If the drug passes phase 2, meaning it is both safe and efficacious in a small patients group, can we sell it in the market? The answer is no, not yet. We cannot sell that drug. Why? Because we have tested only it on few hundred people, which is not enough to be certain about its effect on large population. So at this stage, we only have a hint that the drug may be effective, but we have not confirmed it yet. To confirm, we move to the phase 3. So phase 3 is a very important phase of a clinical trial. It is called as a pivotal clinical trials. So phase 3 trials play a pivotal role in determining whether the drug is truly effective. If the drug passes phase 3, it is considered highly reliable. So some important features of phase 3 is that patients are recruited in the thousands in number. You can see in phase 2 we have recruited only 20 to 200 patients. In case of our phase 3 trials, we recruit in thousands. And in phase 2 trials, we have taken patients from only one location. The study was only unicentric. But in case of phase 3 trials, patients are taken from multiple locations, called as a multicentric studies. Sometimes even we can take patients from different countries. And this ensures that drug is tested in people with different genetic backgrounds. So the main aim of a phase 3 clinical trial is to confirm the efficacy that was observed in a phase 2. So here is one question. In phase 3 trials, why we are taking more number of patients and we are taking patients from the multiple locations? What is the reasons? The reason is that if drug works in thousands of patients, it is more likely to be effective in general populations. And we are taking patients from different locations, and that helps to cover our genetic variations, ensuring that drug is effective across the diverse populations. So that's the reasons we are taking a more number of patients and from multiple locations in phase 3. So what is the difference between phase 2 and phase 3? The difference is that phase 2 provides an indication of efficacy, whereas the phase 3 provides the confirmations of efficacy. So this is the main difference. So after phase 3, we have strong confirmations that drug is both safe as well as effective in a large population. So what happens after phase 3? Now the drug has been tested on thousands of patients. Can we sell it now? The answer is still not. Still we can't sell. The reason is that because although we have confirmed the drug is effective, it is working in thousands number of patients, we need an approval from the licensing authority. So what we will do, at this point, the drug company will submit an application to the regulatory bodies. like for USA FDA, for India DCGI, for Nepal DDA. So this application includes all the data from the phase 1, phase 2 and phase 3 trials. So if NDA means new drug applications. So if NDA is approved, the drug can be finally be marketed. Now we'll see a phase 4 trials. It is also known as a post-marketing studies or a post-marketing surveillance. So here is one question for you. Once a drug is in the market and being sold, does testing stop? The answer is no. We continue to conduct our phase 4 trials, and it is also called as a post-marketing surveillance or post-marketing studies. So why it is important to conduct a phase 4 trials? The simple answer is that to identify the rare side effect and to identify a long-term side effect. This is the main reasons we conduct a phase 4 trials. For example, some side effects are very rare and occurs in only 1 out of 50,000 people. So this cannot be detected in phase 3, which has only recruited 5,000 patients. So this is only possible to detect when a drug is given to the millions of people. As I told that phase 4 trials are done to identify a long-term side effects. And if we see the from phase 1 to phase 3, the clinical trial are for only for short term, lasting only for a few months or maybe for a few years. But some side effects appears after long-term use, like maybe 5 to 10 years. So this can be only observed or possible to see the long-term side effects when the drug has been used for a long time. So, let's take one example. Once upon a time, there was a rofecoxib, and this was developed as an alternative to aspirin because the aspirin caused the peptic ulcer. But rofecoxib was found to be effective and safe in phase 1, phase 2, and phase 3 trials, and it was marketed and used by billions of people in the world. So, however, phase 4 reveals that it increased the risk of a heart attack. So, this drug was withdrawn from the market. So, this can be possible only from the phase 4 trials. Long-term side effects or rare side effects only it's possible when more number of people are using this drug and it is used for a long time. So it is essential for this purpose, and each and every company should do a post-marketing surveillance or a phase 4 trials. So let's talk about phase 0 trials, and phase 0 trials are also known as microdosing studies. So phase 0 trial is a special type of clinical trial and conducted before a phase 1. So it is called as a microdosing studies. So here is one question: why we need to conduct a phase 0 trials? As you know that for developing a drug is a very expensive and pharmaceutical companies should spend nearly about billions of dollars, and we have seen that many drug fails in a phase 1, phase 2, phase 3, and that may cause a loss of a huge amount of money. So phase 0 help to identify the failures early, so money is not wasted on ineffective drugs. So for that purpose, the many companies used to do a phase 0 trial to save their money. So let's talk how does this phase 0 work. So what we do here, a very small amount of a dose, that may be a subtherapeutic dose, is given to the humans, and dose is about a 1 by 100th of the expected human dose. That's maximum of 100 micrograms. So in this case, what we will do, we will use a radio level tracking system. So they will study the way the drug goes in the body, whether the drug reaches the target site or not, whether the drug accumulates in unwanted areas or not. So that can be seen in this 0 phase. So here is one question: what phase 0 trials cannot do? The simple answer is that they cannot determine the safety and they cannot determine the efficacy. They only provide the information on a drug distribution in the body. So this is the reasons. And the phase 0 trial are not a compulsory. It is not a mandatory that all the pharmaceutical companies should do the phase 0 trials. So this is optional and not required by the FDA or a DCGA or DTA. So pharmaceutical company conduct this trial to save their cost and by eliminating non-promising drug early. So this was the end of our clinical trials of our different phases: phase 1, phase 2, phase 3, and phase 0. So let's have a quick revision with some multiple choice questions. Let me ask first questions: which is the first phase conducted in human? The answer is phase 1. So next questions: which is the first phase conducted in a patient? That is a phase 2. And next questions: which phase is the efficacy first checked? Phase 2. In which phase is the maximum number of patients recruited? Phase 4. Which phase is conducted after the marketing? Phase 4. Which phase detects rare side effects? Phase 4. Which phase has a highest failure rate? Phase 2. What is the main goal of phase 1 trial? To determine the maximum tolerated dose, MTD. So what is the main goal of phase 2 trial? To test the safety and efficacy in the patients. What is the main goal of phase 3 trial? To confirm the efficacy and safety in a large populations. So guys, this was the end of our clinical trials chapter. If you like this video, kindly do subscribe my channel, and you can share with your friends. Thank you for watching this video. Have a good day.